So genomics is the study of the order of the bases in a genome. So sometimes we need to know the order of the bases for the first time. Sometimes we already know the sequence of the genome and we want to look for differences. We also use genomics to look at the sequences of pathogens. So for example, to follow the epidemiology of infection. We also use genomics not only to look at order of bases, but to count molecules. And perhaps that's one of the most common uses of genomics at UCD. So the focus of the core is really driven by the instrumentation, and the core is very well equipped. So at one level we have equipment to measure gene expression using PCR or RT-PCR. So we have large-scale equipment that will measure hundreds of samples at the same time. On the other hand, we have equipment that looks directly at the genome. So we offer Illumina Sequencing, so that's probably the core genome sequencing facility. At the moment, the machine that's there is Illumina based. It'll offer mid and high throughput. Will give you data around 800 million reads up to 120 gigabases. However, in the near future, that's going to be even further upgraded, so we'll have an even higher output. We also have single cell sequencing, so we offer 10x genomics, and that allows extraction and making libraries from single cells of organisms. We also offer long read sequencing, so for example using MinION from Oxford Nanopore. And that's extremely useful because it's very simple, very straightforward. So the team from the genomics core can offer a service, so you can provide your molecule or your cells and they will run the material and analyze it for you. Or more importantly, they can get you to the level of a trained user very rapidly, and that way you can run the machines yourself.
So our research is trying to understand how epilepsy develops and then try to, with that understanding, try to develop new and better treatments for the disease. So with genomic and transcriptomic technologies, what we're really trying to do is to identify altered gene expression patterns in epilepsy, to really understand how that altered gene expression may drive the formation and the maintenance of the hyperexcitable brain. So the facility has really helped our research approaches in several different ways. So firstly, training of our staff on the technology and the equipment that is available. The technical staff have really helped us with the design of our experiments and really helping us to get the most out of the experiments that we're performing. And they've been really helpful as well with troubleshooting. So if anything has gone wrong, the facility has really helped us to identify where the problems might have been and to correct those problems. So the benefits for our research in terms of working with the facility and the team has really been the peace of mind, just knowing that when we're doing these experiments, these large sequencing experiments, that there is expertise behind us and support for us with those studies in case we need that.